right, guys, we're picking back up in the next section of our notes, concepts that really we've talked about, but it's time to actually write them down. So Roman numeral 2, passive transport. We had already in the previous lecture talked about diffusion and facilitated diffusion, and now it's time to write down some stuff about osmosis. So first, what is it? Osmosis is really the movement of a solvent, and most solvents are water. So we see the movement of water down a gradient, down its gradient, which is why osmosis falls under the umbrella of passive transport. Because passive transport is any time a substance, be that solute when we were talking about diffusion and facilitated diffusion, or now solvent as we were talking about water. So the movement of that substance from an area of high concentration to an area of low concentration down its gradient. So again, this second bullet really is in writing what I just verbally said. This is still passive transport because water moves from high to low water concentrations. And really the reality is it's all about the movement of something called free water. So anytime you have, I'm going to actually take that away for just a second. Anytime that you have a solution with solutes in it, so these little blue dots, solutes, every single one of those solutes is attracted, as long as it is polar or has some kind of charge, it's attracted to water. And so the more solutes, like the sodium, the more solutes you have in a solution, the more water molecules will be attracted to each individual solute. And when a water molecule is attracted to a solute, it's locked into something called a hydration shell. That means that the water molecule is kind of bound to that solute and it is not free to move. And so when we talk about osmosis, it's really predicting the movement of free water, so the water that is not bound in hydration shells, which should or could maybe make sense why water concentration is highest in areas where solutes are lowest, right? So free water is more available in hypotonic solutions. And so when we look at water movement, the water typically will move from a hypotonic environment into a hypertonic environment, one with a lot of solutes. I've said before in sort of our class times, water chases after solutes. So it's always going to want to be attracted to the area with the highest concentration of solutes, which of course you know is the hypertonic environment. So when we talk about tonicity, we need to make sure that we understand or remember that using words like hypotonic, hypertonic, isotonic, that terminology is in reference to the solutes that exist in a solution. So if we have high counts of solutes, it's a hypertonic solution. If we have low counts of solutes, that's a hypotonic solution. And if we have solutes that are equal on either side of a membrane or equal in two different environments, then we see an isotonic solution. And so here it is in writing, what I've said multiple times. Water always chases after solutes. So when osmosis occurs, water is always going to choose to move into the hypertonic environment, the one with all the solutes. It is attracted to all of those solutes. Now for the next section of notes, you see that it says plants and animal cells. I want you to build out this chart in your notes before you start moving forward in the lecture video. So we need to talk about what do plant cells and animal cells do when they are placed into hypertonic, hypotonic, and isotonic environments. And again, you won't be surprised that this connects back to the structure of plant and animal cells. So build this out in your notes. You can pause the video if you want, and when you're ready, come back to this. I'm going to move to a Jamboard so that I can show you some pictures and also diagram a little bit. So if you're back, you have created this chart: animal cell, plant cell, hypertonic environment, hypotonic, isotonic environment. There's some funny words down here, which I'm going to kind of drag and drop, vocab words that refer to the state of a particular cell once it is placed in those environments. And then I've also given you some pictures. So the example that we're using here in all of the animal cells, these are red blood cells. And then down here you see sort of that rectangle shape with the big watery vacuole in the middle. And so this will represent our plant cells. So we're going to draw a little bit, and then we're also going to label a little bit. So first, let's draw what's going on here. So in your box for an animal cell placed in a hypertonic environment, we're going to draw, let's see if I can do this with my finger. We're going to draw a beaker. Woo. There's a beaker. And then here's our little red blood cell inside the beaker. Now, this is an animal cell or red blood cell placed in a hypertonic environment. So I'm going to make this a hypertonic environment, and you should too. So we're going to put lots of little solute dots out here in the solution as best as I can. Right? So knowing that water chases solutes, think about if I dropped an animal cell into this super hypertonic solution, which way would water move? Ooh, that's a mouthful. Which way would water move? So think about, is water going to go out of the cell or is water going to come into the cell? Well, water is going to chase after all of these solutes and it will exit the cell. And so what will that cell ultimately look like? This picture right here shows us. This is a normal red blood cell for perspective. Here is one that's a bit shriveled. And so a fancy term for this and what you should add to your chart, let's put it up here, is crenated. So when animal cells are placed into hypertonic environments, they lose a lot of their internal water to the hypertonic environment and they sort of shrivel up or they become crenated. Now, this can ultimately kill an animal cell if the environment isn't restored, if water balance isn't restored. So let's move on to the next animal cell situation. So I'm going to draw my beaker again, and here's my animal cell. And so this time it's a hypotonic environment, which means really there's very few solutes. I'm going to make them smaller this time. There's very few solutes out here, but there's a whole lot of solutes inside the cell. Ooh, this is a challenge. Inside the cell. And so think about what will happen in this situation. Which way will water move? Water chases those solutes, and so water will rush into the cell. And this is the result. The cell begins to swell, and eventually, if water continues to rush in, because of the lack of a structural molecule around the cell, that cell's going to explode, basically. It's going to rupture. And so the word that we use here, we say that the cell lyses or the cell has lysed. That means to split or rupture. And so our last scenario is what happens in an isotonic environment. And so here is our beaker again. Here is our cell. And isotonic is same number of solutes inside. So let's see, I'll put 2 solutes inside and 2 solutes out. What happens to water? Now keep in mind, we talked about water doesn't stop moving, but it will, in an isotonic relationship, move into the cell and out of the cell at equal rates. And so this is the preferred situation for an animal cell. In this environment, that cell would be considered normal. And this is one of the reasons why when you, if you've ever been to the hospital or know anybody that's been to the hospital that has been hooked up to an IV for hydration, they're not pouring or running tap water into your blood. That would be terrible. That would be doing this. That would be putting all of your slightly salty blood cells in a hypotonic environment, and they would rupture. And so instead, they run a little bit of a saltwater solution as a part of an IV fluid, and that is going to match the tonicity of your cells, so that water goes into and out of your cells and keeps your blood cells nice and healthy. All right, let's look at a plant cell. So in a plant cell, let's go back to drawing here. In a hypertonic environment, I'm going to do this. So we have our plant cell right here in the same kind of environment. And so here is same as the animal cell. We've got lots of solutes. What is actually going to happen here? Well, the exact same thing that happened with the animal cell. The water is going to move out. It's going to chase after those solutes and move out of the cell. But check out this picture. The overall plant cell doesn't appear to be shriveled. It stayed essentially the same shape. And so think about why in the world might that occur? Because it has a cell wall. So it has this nice, tight sort of structural boundary that maintains its shape. Look at the inside, though, of this plant cell. Here's the central vacuole. There's our shriveled concept. The central vacuole is not holding much water. And so if I put this under a microscope and could really look at the inside of the cell, even though it appears to maintain its shape because of the cell wall, all of its internal contents would be shrunken down into kind of the middle of the cell. And this sort of a plant is on its way to dying if it's not corrected. So these cells are called plasmolyzed cells. Plasmolyzed cells. All right, so let's go back to drawing for a moment. I like this one. Here is our beaker again. Here is our plant cell, and we're going to put it in a hypotonic environment. So same thing as the animal cell. I'm going to put a few solutes out here showing hypotonic, but then for perspective, we're going to load the inside of this with lots of solutes so you can see why the water is doing what it's doing. So in this case, water still chasing after those solutes will rush into the cell. So the cell gaining water basically is storing that water in its central vacuole. Because of the cell wall, though, the overall cell does not rupture like we saw with the animal cell. And so this is actually a preferred state for a plant cell, and it is called a turgid cell. Last one, if we put our plant cell in an isotonic environment, so here's our beaker, here's our plant cell. If we have same number of solutes, we have water moving into and out of the cell at equal rates, which might seem like or feel like this is what is normal, this is balanced, this is healthy. But actually remember that that central vacuole actually likes to be nice and full and swollen to put a little bit of pressure on that plant cell wall. It helps with the movement of water throughout a plant's tissue. And so although this was normal and good for the animal cell, this is actually flaccid or a bit wilted for the plant cell. So jumping back over into our notes here for a moment, I want to make sure that you captured this last little bit, kind of walkaways from the chart. So important concepts, just write this below the chart that you diagrammed. Important concepts from the chart: plant cells like to be slightly swollen or turgid, which only happens when they are placed in hypotonic solutions. So plant cells actually like tap water or tap water-like solutions, which is why it's perfectly fine if you have indoor plants and you water them with your tap water, great. That actually is allowing water to move into the plant cells. If a cell has a cell wall, it won't rupture. And so we saw quite a difference in the end outcomes of plant cells as water moved. And last but not least, I want to make sure that you catch that an animal cell actually prefers an isotonic environment. So it is looking for an environment in which water is moving back and forth at equal rates. Last one. Plants prefer hypotonic. So we kind of talked about that with the first bullet, but there it is, nice and plain. There's a plant cell's preference. All right, when you are done with this, you're done with the notes, and you'll be ready for the next time that we meet together.